So first of all, thank you everyone for making it the last the last talk of the day. Maybe I don't know about you, but I feel like maybe we just took like a quick deep breath and just kind of like inhaled and exhaled. just to get ourselves back in last last talk of the day. Okay, so I'm gonna go ahead and share my screen now. This way. Okay. All right. All right. So, Bayesian Metabolic Control Analysis for Fungi. That's the title of my talk. And basically, I want to, first of all, just acknowledge all the folks who've been involved in this project. Shant, Peter St. John developed the algorithm. Andrew McNaughton is now a P&L employee who transferred the technology to P&L. James was an intern who helped with that. Joseph Niraj funded this. All the folks who have developed the software, who've performed the measurements, and of course John Magnuson and the Agile BioFoundry team. This was really funded by the Agile BioFoundry, and then we've had some LDRD funding to extend it further and apply it to other organisms. So I think the first thing I just want to mention is what's the point of Bayesian metabolic control analysis? What is Bayesian metabolic control analysis? And so you have to imagine that you are trying to design a cell factory that can produce a compound that is of interest to industry, and you know that in order for it to be economically viable, you have to be able to achieve a certain level of titers, rates, and yields. So given so much glucose in, how can you get your a certain rate or a certain titer of your product out? And we have the ability to develop, we have the ability to measure multiomics data, and we have prior knowledge of the metabolic network. And the question is, how can we use this information to perform some kind of sensitivity analysis that would allow us to predict, to say, all right, well, if I were to increase the expression of this gene or decrease the expression of this enzyme, that would affect my titers in some way. That would improve the titers, rates, and yields. And so that's the main idea behind Bayesian metabolic control analysis. Bayesian because we're taking into account all the uncertainty that comes from measurements, and also there's uncertainty in the sensitivity analysis itself, and we don't know exactly what the kinetic parameters are, but we can learn from data, and so we have prior knowledge which we can represent as a prior distribution, and given some evidence, we can hope to find a posterior distribution that gives us tighter bounds on our beliefs. And it's called metabolic control analysis because that's the sensitivity analysis part. It says basically there's two kinds of information. There's an elasticity, which tells you how sensitive an enzyme is to a change in a metabolite level. And then there's a flux control coefficient, which tells you how sensitive the product you care about is in how sensitive it is to a change in enzyme level. And it's a system-wide parameter, so all the enzyme flux coefficients sum to 1, so you can kind of rank order your enzymes to say this is the one that is most, this is the one that my titers, rates, and yields are most sensitive to, and this one is second most sensitive to, and this gives you targets for predicting what experiment or what strain I should develop next. So that's Bayesian metabolic control analysis in a nutshell. If you got that, then you can, that's the main thing. And so I'm just going to show you in the rest of this talk. I'm going to show you how we applied this to 3 different organisms, 3 different fungi, And then I will, if there's time, I will take you through a demo which explains how this works on a very small pathway in detail. And I'll be taking you guys through a Jupyter notebook so you can see, exactly what's involved in performing that analysis. But just to recap, the learn goals for microbial cell factory design is to identify what native or non-native enzymes have the largest impact on titers, rates, and yields. And we're just using knowledge of the genome scale metabolic network and multiomics measurements, and we want to quantify uncertainty in our predictions based on uncertainty in the data. So the way that Bayesian Metabolic Control Analysis works is its inputs are multiomics measurements relative to a reference strain. So you have some fastest growing strain, the one that's producing the most of your product, but you have a whole bunch of other strains as well. And you want to use all that information from the slower growing strains to perform that sensitivity analysis on the reference strain. And so you can take measurements, multiomics measurements, both proteomics, metabolomics, fluxes, any of that kind of information. All that information is useful in calculating these flux control coefficients, or the which tell you which enzyme perturbations are most likely to improve titers, rates, and yields. And so the assumptions, we make a steady state assumption, and that constrains the reaction fluxes. We use Lin-log kinetics to predict reaction fluxes for metabolites and enzyme measurements. And we use the knowledge of reaction stoichiometry to generate prior beliefs about the effect of change on each metabolite to a change in a reaction flux. So how does a particular reaction, how sensitive is a particular reaction to a change in a particular metabolite? And that elasticity tells you that how sensitive it is. If it's negative, that means it goes up. If it's positive, it means sorry. If it's negative, it means that as you increase the metabolite, then the flux goes down. If it's positive, it means that as the metabolite goes up, then the reaction goes up. And so, but there's some limitations to it. One is that your input perturbations are restricted to just changing out the nutrient media, or perhaps the overexpression or repression of genes. You can't fully knock out a gene, and you can't insert new genes. The system doesn't quite deal with those changes very effectively, and so you have to kind of restrict yourself to the kinds of perturbations that you can actually do. Okay, so the question is, how do we specify the elasticities, and what prior knowledge can we use to at least come up with a good guess as to what that elasticity could be, even in the absence of data? And so one piece of knowledge that we can use is knowledge of the metabolic network. So here on the left side here is a reaction network. Reaction A takes this blue dot. Reaction A takes this blue dot, and this blue metabolite produces this orange metabolite, M1. And M1, as M1 increases, well, it kind of, just from normal thermodynamics, we expect that this reaction rate will slow down as M1 increases, as M1 increases in concentration. So you can think of this as having a negative or inhibitory effect on this reaction. At the same time, M1 is a product of RB, and so the larger this concentration, the faster this reaction rate will go. Those are our intuitions. And then it could also be the case that M1 has some kind of regulatory effect on RC, but we don't think that that's true in general, so we're just going to assign a very small a prior distribution to that. We say basically we'll allow for the possibility that M1 can inhibit or activate RC. We don't know, but if our priors say it's pretty much close to zero, but we allow for the possibility that data could change our mind. But the idea is that because RA inhibits RA, so M1 inhibits RA, then we say that this product inhibits the reaction rate. So we have a negative half normal distribution, and we don't know what the true elasticity is, but we think it's less than zero. And likewise, the effect of M1 on RB it's a reactant, and so we believe in general that reactants activate the reaction rate. And so we don't know exactly what the elasticity is, but we think it's greater than zero. And lastly, we have this regulator elasticity, and we just assume that in most cases, most metabolites don't inhibit or activate other enzymes that they're actually not part of, but we allow for that possibility. Okay, so what do I mean by Lin-log kinetics? So let's assume we have some reaction A goes to B plus C. Then we can model the reaction rate as, so the V is the flux here, the reaction rate, and that's equal to the enzyme concentration times a constant plus some parameter A, we don't know what it is, times the log concentration of A, plus B, some other parameter, we don't know what it is, times the log concentration of B, plus C at a third parameter, we don't know what it is, times the log concentration of C. Now, because B and C are products of this reaction, we can at least say that B and C are probably less than 0, and we can say that A is greater than 0. And well, then the question is, why use Lin-log kinetics? And I guess the point is that it turns out it's, for computational reasons, it's much more efficient than trying to solve a Michaelis-Menten equation. On the other hand, as long as you're nearby your reference state, then you actually can you're pretty indistinguishable from a Michaelis-Menten curve as long as you're near that reference state. As soon as you start getting further away from it, this is a logarithm, so it goes up to infinity, whereas Michaelis-Menten will hit some Vmax. So far from equilibrium, it's not very accurate, but at least near equilibrium, near the reference state, it seems to be pretty good at modeling Michaelis-Menten. Now, let's just see what happens when we apply. So that's it. So then the only thing we do now is we give this equation actual data, and then we compute posterior distributions on A, B, and C, these elasticities. And once you have those elasticities, once you have a posterior distribution on the elasticities, you can compute that flux control coefficient, and that rank orders all your enzymes. And so what I'm going to show you is examples where we applied this metabolic control analysis to 3 different fungi. And what will be interesting is how well the algorithm does at calculating a posterior distribution with a large amount of data, with a small amount of data, and then something in between. So the first one I'm going to show you is what happens when you have just gobs and gobs of data. So this is yeast. We have 203 metabolites. We have 240 reactions. We have 5 different media conditions. We have 1,800 metabolite measurements. Of those 203 metabolites, we have 792 boundary flux measurements, and we have 480 enzyme measurements. And so when you do that, then at least in that region that I told you where the Michaelis-Menten is not too where you're nearby your reference state, we actually do a pretty good job of predicting the metabolite concentrations. Likewise, the flux and the proteomics. As you get further away from the reference state, then we don't do as well, but that's expected, because we do believe that Michaelis-Menten is really governing these equations. So this is just showing you what the because we're developing a model, we can not only predict, we can not only we can predict the metabolomics, and we can compare them against actual measurements. Okay, so then what is the advantage of this? Why couldn't we just do a correlation analysis between enzyme concentrations and our metabolite concentrations? Just the correlation between like our rates. We can just take a look at our bioproduct rate and compare that, and see how well that correlates with enzyme activity. And presumably the enzyme activities that are most correlated are the ones that we should be adjusting. And so what you see here is a correlation plot between enzyme concentrations and all the boundary fluxes of this yeast model. And you see many different candidates. There's lots of positive correlations. There's lots of negative correlations, and it's really hard to figure out which is the right target to try. You have so many different enzymes to try. Whereas when you calculate these flux control coefficients, basically it gets rid of these spurious correlations, and what you're left with are much more likely to be causal. And so in the same sense that these flux control coefficients tell you which enzymes to perturb, it only gives you a small number of candidates in comparison to just doing a correlation analysis. So that's kind of the best case scenario when we just have gobs and gobs of data. And the question is, what happens when we don't have so much data? And so this is a Bayesian inference for integrating Yarrowia multiomics data sets with metabolic modeling. And we tried this. There were two data sets that we considered. One of them was a 9-sample multiomics data set, which was basically 3 strains in triplicate. And the other one was a 24-sample multiomics data set, which were grown on 2 different sources, glucose and malate, and that resulted in 24 samples with 4 strains in triplicate under 2 different conditions. And so we can see that there was, that we were not able to predict very well protein expression or metabolite concentrations beyond a very small range. But we still had issues even when we were doing this for the 24 sample data set. So maybe that wasn't quite enough. And likewise, we see here that so those flux control coefficients that I was telling you about, it's so the gray here shows you the prior distributions on those flux control coefficients, and the red ones show you the posterior distributions on these flux control coefficients. And again, it's kind of hard to say. There's a few candidates here under the 24 sample that kind of really stood out. There's also some ones that stood out in the 9 sample, but we're not sure if we really believe them. So this is something where we kind of have to go to the experimentalists and say, okay, well, we've got a bunch of predictions. We're not super confident in them, but maybe some of them will yield an answer. So then the third situation is where we had Aspergillus niger producing 3-HP. And again, we want to identify which native or non-native enzymes have the largest impact on 3-hydroxypropionate. And we're using the knowledge of the genome scale model, the multiomics measurements. And in this case, we have a whole lot more data. We have extracellular flux measurements, we have metabolomics measurements, we have extracellular metabolomics measures, we have intracellular metabolomics measurements, we have global proteomics, we have targeted proteomics, and we have 17 strains that are in triplicate. So we have like 54 strains that we can apply to this. So we have multiomics measurements on every single one of them. And then in addition, we also calculated intracellular fluxes. We had extracellular fluxes, but from the extracellular fluxes and the global proteomics, we could use that data to come up with condition-specific fluxes. When we do that, then we have even like an additional data source with which to constrain our Bayesian metabolic control analysis. And so when we do that, and this was the engineered pathway for 3-HP, basically starts from pyruvate, ends with 3-hydroxypropionate. There's a few other routes to getting it, but in this strain, we were just considering this one pathway. And here you see that for most of the cases, there really wasn't a strong flux control coefficient, but in these cases, ALD6, this reaction 118A, and these reactions over here, we had very, very strong flux control coefficients that we had a lot of confidence in. And so then John Magnuson and his team went back and validated those. And so we're still waiting to get the results of those, but we're waiting eagerly because we have a lot of confidence in these predictions. And again, if we compare the correlation coefficients of 3-HP versus all the different enzymes, then we do see several reactions that had pretty high positive correlation and those that had very strong negative correlation. And what's interesting is that hardly any of these ended up being the most important, the ones with the highest flux control coefficients. And so now we have a negative test set. And so we can go ahead and try to knock out these guys or overexpress them, and see if Bayesian metabolic control analysis was just wrong. And in fact, the thing you're better off doing is doing enzyme correlations with your metabolites. And so we have a negative set and we have a positive set for John Magnuson to try to see if it's going to really work. So so we had all sorts of enzymes that we thought were top predictions, and we're going to see if that's actually true. And this was a part of the Agile Biofoundry, which is like a multinational lab consortium. And now, if you guys are still up for it, I thought we could at least take you through a metabolic control analysis of like a very simple pathway. If you guys are still interested, it's what, 8:13 right now. Do you guys? Maybe I should stop and just ask if you guys have any questions. And then if you guys are still up for it, then you're still interested, I can take you through the demo.
I did have a question. You mentioned boundary flux measurements.
Yes.
I'm not familiar with what technology gives you boundary flux measurements or what it's measuring.
Sure. So all you're doing is you're measuring extracellular metabolites in time course. And
Okay,
so
great.
you say, all right, I measure the extracellular fluxes at time 0, time 1, time 2, time 3, and then I just basically draw a line, a slope, and that slope is the boundary flux.
Okay.
Is basically how much that the metabolite is either increasing or decreasing in its rate.
I see. So the boundary being outside the cell.
Correct.
Okay. Got it.
Other folks with questions?
I
Yeah.
have a question about kind of in general. So I guess, are there any like things people need to be aware of when they're kind of like doing their experiment design, or in terms of like, the amount of data that you needed to have a high confidence kind of prediction in this case?
Yes. And so I think the reason why I showed you the Yarrowia was because that was one that was kind of underpowered. And as a result, you got a lot of kind of weird looking predictions and things. It was very clear that it was either over constrained or under constrained, but it was not producing the kind of expected behavior that we want from our Bayesian metabolic control analysis. And that can happen if you just don't have enough data to well, actually, if you don't have enough data to do anything, then basically what you end up with are your prior distributions. And that's basically just using your prior knowledge of the metabolic network, and that's all you can do. So hopefully your posterior distributions are tighter than your prior distributions. But if your data is like all over the place, then it may end up that you have more variance in your posteriors, which could happen, right? So that's one thing, is you got it. So it looks like 9 samples is definitely not enough. 54 samples is definitely enough. Or certainly, I think the yeast one had 5 samples with a bunch of different nutrient conditions. I forgot to actually calculate how many samples it was involved, but it had a lot of data associated with it. It was very deep measurements, and you're talking about yeast, and so that organism is extremely well understood. And I think they had absolute concentrations. I mean, they just went nuts, right? One of the advantages of a metabolic control analysis is that it does not require absolute concentrations of metabolites nor absolute concentrations of proteins, because everything is relative to a reference strain. And so as long as you kind of did it all in the same batch, because of course there are all sorts of batch effects if you try to measure one proteomics data set with another proteomics data set that came from a different batch. But as long as they're all part of the same batch, then it's very comparable, and so that kind of data works really well. Other kinds of limitations. Well, I was going to show you some of those issues that can come up, especially in the inference in the demo. And so there's ways of determining, did my inference actually converge, or do I need to kind of adjust the model in order to fix things? And so there's some tweaking that can occur if things don't go as you would like them to go. And so there's diagnostics that tell you how to treat and how to detect and treat those issues.
Cool. Just a couple of things I want to clarify. So So you mentioned that, the metabolic data, either it's proteomic or, metabolomic, it needs to come from the same batch. So there is no good way to kind of eliminate like batch effects, at least for, your model at this point.
That's a really great question. Batch effects is kind of a catch-all for basically everything that can go wrong between two different experiments, right?
Right.
And so and so I suppose if we knew who the operator was and what instrument that they were working on, and whether it had been cleaned earlier that day or if
Right.
it had been cleaned last month, then we might be able to take that kind of information into account. But we don't have that information, right? So without those are all kind of in some sense unobserved confounders, and so it can be very misleading to try to use data from two different batches when there are very clearly important batch effects that can occur. Yeah.
So there's, I guess, because I think there are some like sometimes people do some kind of, like zero mean, like, normalization to trying to eliminate
Yep.
or like at least reduce batch effect. But
Yeah.
I guess for the Bayesian kind of prediction that you're doing, it's still like way too much noise to, for it to work well. Is that the,
It just depends on the data set. Like I
yeah.
was dealing with some mouse data, or I think it was just cell culture data. And in that case, I was taking a look at mock versus infected, right? So either this, cell culture has been infected with a virus or it hasn't, right? And it turned out that the mock versus infected in one batch was closer to each other than the mock versus mock in the two data sets,
Mm,
right? So, in that case, you really can't use it.
right.
But
Right.
certainly there could be cases where, if you could, maybe you could control for the variance if you know what you're doing.
Okay. And my last question, sorry.
Sure, no, this
Last
is
question I
great.
have is like for, I guess in terms of, experimental design, like would you want to kind of design your experiment to produce or yeah, in whatever case that you can do to have more variation of, like variation in your like data, if, like in your example, like between mock and like the infected, would you like would that like variation, like large like differences that you see can help you have a better prediction?
So there are definitely analysis methods that work well when you have very kind of
Yeah.
different data. Bayesian metabolic control analysis is much more subtle. It's basically like you're
Okay.
just trying to figure out how to increase it from 22
Mm-hmm.
grams per liter to like 25 grams per liter,
Right.
right? So you're just like nudging
Okay.
the network in just the right direction so that it'll bump up the titers rates and yields.
Mm,
And so it's very easy. You can imagine it's very easy to hit like a local maximum, and in which case you might have
yeah.
to do something a little bit more extravagant to kind of push things into a
Right.
different valley, right?
Yeah.
Into a different valley. So this is kind of like a very fine. This is like when you've tried everything else, all the things you could think of. Now you throw in Bayesian metabolic control analysis just to get it over the edge, right?
Okay. So it's actually kind of designed to capture like smaller changes.
Yes.
Okay. I see.
Yeah. Yeah.
Okay. Thank you.
Yes. All the way in the back.
So how does this kind of compare to flux balance analysis if you're trying to get a optimal metabolite, like the optimal rate or a nudged rate in a better direction of a certain metabolite?
Yeah, yeah. So flux balance analysis actually this uses flux balance analysis underneath the hood,
Okay.
but unlike flux balance analysis, it can actually use proteomics, metabolomics. And it can use proteomics data and metabolomics data from multiple different strains, right? So in FBA, you can have, um, for example, I can constrain one particular set of fluxes. Um, with proteomics data, right? So eFlux is an example algorithm that does that. We published that back in 2006. IMAT is another. In eFlux, it tends to work really well for if you're trying to do microbial cell factory design because it puts hard constraints on the fluxes. given changes in expression data. And there's another algorithm called IMAT, which uses soft constraints. And that's actually better if you're trying to tell a story, because it basically tries to make the fluxes as consistent with the proteomics as possible. And so it tends to work well when there's, when you're like dealing with very complex, human-like or mammalian data, and you're like just trying to figure out what's going on. You can tell a story much more effectively with IMAT. But it's not very good. But its fluxes tend to be so it's good for telling the story. It might not be so good for actually trying to improve titers, rates, and yields. And so that's the issue, is that I think Bayesian metabolic control analysis, of all the methods I've seen, really does very, very well at using the data and combining with prior knowledge to get a suggestion as to what to do next.
Gotcha.
Whereas, yeah, so FBA can do things. You can do things with FBA like large scale changes. Like if I knock this gene out and I added this other gene, how would that affect my titers, rates, and yields? And so those are some large scale things. But for the fine tuning, Bayesian MCA makes the best use of the data, right?
Awesome. I have actually one more question as well.
Go for it.
So you were saying to evaluate the data from the related organisms, or I guess the organisms that we're producing, as much metabolites as the one you're trying to model, or I guess I don't know the right word for that. But why would you pick the lower organisms, or like the less performing ones?
Yeah. So I think it actually goes back to this figure here. Not this one, but you're trying to calculate a slope, right? And so if I only have data at the actual V star, then I don't know. I need all that other data to kind of parameterize this slope, so that this Lin-log curve, so that it matches the data. And so I need data beyond just the reference in order to be able to figure out what that slope actually is.
Gotcha. Okay.
Yep. So I'm looking like, what happens if I change the external metabolites, increase them or decrease them? What happens if I overexpress a gene or repress it? And so these all create perturbations that allow me to kind of do that sensitivity analysis around the reference state.
Gotcha.
Cool. So we have 5 minutes left. I don't know if I can really do a demo justice, but I can at least point you to the code so that you can actually download it and run it yourself if you're interested. So let me just go ahead and go there. So if we go to GitHub, github.com/pstjohn/EMLL. That will get you there. And I'll just, and so this is the ensemble model with linear logarithmic kinetics. And if you follow these directions, you should be able to get it working. And then there's a notebook. There's actually 3 notebooks. I was going to show you the Wu 2004. That's a very simple linear pathway. But there's also one that shows it on a medium scale model. And then the Contador shows you on like a medium scale model, and Hackett shows you that yeast model that I was telling you about, where he just threw everything at that, every measurement that's ever been made on yeast, basically, at that data set. And the cool thing is it scales to those to almost genome scale. So it takes a little longer, but you have to give up some. So on the small scale models, you can actually run Markov chain Monte Carlo, or Hamiltonian Monte Carlo, which kind of is guaranteed to converge to the true underlying posterior distribution. But when you start to get to these larger scale models, then you have to use something called variational inference, which is a very fast method. It's a gradient descent, but it tends to underestimate the variance because it's not taking into account all the correlations between all the elasticities. It's assuming that every elasticity is its own thing and there's no joint distribution that it has to worry about. And that assumption is usually not justified, right? So So it tends to underestimate, but it gives you pretty good means, and so it's a good way of at least getting a ballpark as to where you ought to be, and then you can maybe try HMC at the very end to kind of fine tune it.
So I have a clarifying question, Jeremy.
Yes.
This is the aspergillus data that you showed was based upon reactions as opposed to genes. Those are two separate things, and particularly in some fungi, aspergillus is one example. The entire clade does have a lot of repeated regions and repeated genes. You get gene families that do the same reaction. So when you're talking about changing a reaction, it can't really be targeted with a single gene change. It might require multiple engineering events, for instance.
You are absolutely correct. And so in the original version of Bayesian Metabolic Control Analysis, basically, I mean, he was a very, very good mathematician, but he wasn't thinking so hard about the biology, so he was just like one gene, one enzyme. That was true, I guess, with Tatum and Beal. It was Tatum and Beal. Beetle and Tatum, that was it. Beetle and Tatum, right? But obviously that's not true in general. So we have modified, we've extended the Bayesian MCA to take into account the possibility that you have either isoenzymes, so multiple enzymes can simultaneously catalyze, independently catalyze a reaction, or members of a protein complex. And the way that we combine the data takes into account those distinctions. So, but your point is well taken in that you may have a very strong flux control coefficient, but it requires you to modify, 10 genes. That's really not very useful, right? So there's a discussion that needs to take place after the Bayesian MCA, which is, all right, here are our targets. Which ones are actually actionable? And maybe it's not the highest flux control coefficient, but maybe it's the second highest or the third highest, right? And so we have to have that conversation to make sure that we're giving actionable targets to our metabolic engineers.
I have another question, and it was following up on a question that Jason asked, which was about,
boundary fluxes?
Boundary fluxes. Does that take into account, so you said that you assume a steady state. I'm guessing that that's based upon like bioreactor growth conditions. That being that you're taking time points, but does that take into account like biomass increases or just sheer concentrations of metabolites when you're making those measurements?
No, that's a really excellent question. So yes, in a bioreactor, so flux balance analysis was kind of designed for bioreactors, right? Or chemostats, where you can ensure that the flow going in equals the flow going out, and then you can truly have a steady state. Instead, what you have is like when you have batch cultures, which is basically all we can afford to do in the Agile Biofoundry, then basically you're looking at like that log phase growth where you can at least make an approximation, and you're taking into account that at least in that region you can have some kind of reasonable prediction for steady state or quasi steady state, where you're taking into account the growth rate of the organism as well. But yeah, you have to kind of worry about, you don't want to you don't want to get exponential, you kind of want to get log phase. You don't want it to, there's like a sweet spot where you have to kind of get the measurements in order for it to make any sense at all.